Today we shall discuss on polymeric nanomaterials and devices, mostly biomedical devices. Now, you know, today nanotechnology has come to the forefront. Everybody has got interest in this nanotechnology, development of various devices using nanomaterials, which involves nanoscience. You know what is nanoscience? Science of small dimension, nano dimension. So, that nanometer scale science has been devoted to design and construction of functional structures and the utilization in suitable utilization for suitable devices, and that is in biomedical field that nanotechnology is being used for treatment, for diagnosis, for monitoring and control. And this nanomedicine devices contains nanoparticles, nanomachines, nanofibers and sensors. Nanoparticles, you know, there are various techniques to form these nanoparticles or nanomaterials. There are two major approaches, bottom up approach or top down approach. So, if we can make some materials made of these nanoparticles, these particles in nano dimension, say might be below 100 nanometer or 200 nanometer, like that, they show some unusual properties. You know, gold, its color is yellow. When it has been broken down to nanoparticles, gold nanoparticles, its color changes from yellow to blue to green to purple to red, and the properties are different. Okay. Now, you have seen in case of carbon black, there are various grades of carbon black, of which one grade is channel black or sometimes furnace black. Channel black, they are available in very small particle size. Furnace carbon black is also available in small particle size, say 20 to 30 or 40, 50, 100, 200 nanometer dimension. When those particles are used as filler in rubber compounds, we get very good reinforcing properties, improved strength of the rubber composite. If I mention one aspect, say in homeopathic treatment, homeopathic medicine, you know, homeopathic medicine, that is the strength of the homeopathic medicine depends on dilution. Higher dilution is the stronger medicine, and lower dilution means concentrated solutions or higher concentration, solutions of higher concentrations are little lower strength. Okay. What is the why this happens? You know, these homeopathic medicines, these are made from chemical products, either synthetic products or from herbal products, herbal base. Okay. In all the cases, some concentrated medicine is taken and it is diluted, and there is a process of dilution. Not simply you add that medicine in a solvent, say water, and you get the dilution. No, they have certain process, they follow certain process, they give some mechanical vibration and they get higher dilution homeopathic medicines, and that becomes the stronger medicine. Now, in that higher dilution homeopathic medicine, with the help of available instrumental facilities, the presence of that chemical compounds of that medicine would not be detected, but still it is stronger medicine. In case of high concentration medicine, say lower potency, there sometimes one can detect the presence of those chemical compounds. Why this happens? Now, my concept is there might be some sort of separation of the molecules or dispersion or solution of those medicinal compounds in molecular level. In aggregates, they function in one way. In dispersed condition in molecular level, they function in another way. That might be the reason, but that research has not been done, we do not know. But today this nanomaterials or nanotechnology indicates us that by virtue of the small dimension as well as their high surface energy, they are prone to form aggregates, agglomerates. Okay. If we can break the aggregates, then we can get the dispersion in a smaller dimension. Probably we can exploit the real properties at the molecular level. So, there are certain properties at the molecular level. The aggregates of those materials may not perform the same level of properties that we have visualized in some cases. You see, the color of gold changes from lump to these nano particles, and their properties are also changes. So, this concept is being utilized in various fields for various purposes, where also in biomedical field people are trying to utilize this nanotechnology for treatment, for diagnosis, for machines, micro machines, etc., nano machines, okay, as well as in sensors. So, I will give you some exposure to you with this thing, those materials and some devices. Okay. So, the term usually applies into living or processed tissues, biomaterials. It usually applies to living or processed tissues or to materials used to reproduce the function of living tissue in conjunction with them. Simply, it is a material intended to interact with biological system. Say biomaterial meant for using in some biomedical device that is used in place of living tissue, so that that device can function like that of the host tissue. So, it should be biocompatible, it should be non-toxic, it should be bioacceptable, and it should perform the real function what you want. Now, what are those materials for used in implant devices, biomedical implant devices? We can use polymers, we can use ceramics, we can use metals. Okay. Now, you have some exposure to polymers, names of various polymers, various categories of polymers you have seen, addition chain polymers, condensation polymers, okay, and you know their structures. Addition chain polymers exclusively contains carbon chain structures, condensation chain polymers contain carbon as well as other heteroatoms in its backbone structure. Condensation polymers are more polar than adhesion chain polymers, right. And looking at the structure of a polymer, you can predict its properties, you can predict its inertness, its reactivity. Say for example, you want to make a device, you want to make some article of course. If you know the chemical formula, chemical structure, chemical nature of the polymer, you can predict whether it will remain stable or it will be little unstable in the environment or within a living system. So, polyethylene, if I talk about polyethylene, polyethylene is not degraded by environment, it is not degradable by environment, ambient environment, isn't it. If it is exposed to ambient environment, say exposing to sunlight, exposing to humidity, exposing to rain, exposing to air, etc., all these things. So, polyethylene is not degraded, that is why polyethylene is causing lot of problems, nuisance in the environment, because it is a stable polymer having carbon carbon bond and carbon hydrogen bonds, whereas a polyester which contains carbon carbon bonds as well as carbon oxygen bond and carbon hydrogen bonds. Now, this carbon oxygen bond, it is located in the ester group. You know, this ester group is degradable, hydrolytically degradable in presence of acid or alkali, it can break. So, it can be degraded a little bit. Naturally occurring polymers, you know their structures, formulas, chemical composition. Naturally occurring polymers contain carbon, hydrogen, nitrogen, oxygen, sulfur, all these things, isn't it? Majority of them except natural rubber, majority of them contain such atoms as well as bonds are present, and those are easily degraded by nature. Oxidative degradation, thermo-oxidative degradation, microbial degradation, all these degradation occurs over there. But if we put these polymers in living system which contain biological fluid, the pH sometimes is around 7.4, or within the stomach pH may be little lower or higher. So, in those environments, if polymers are put over there, if the polymers get hydrolyzed within the living system, then the stability of the polymer will be at stake. Now, there are certain requirements in biomedical field that we need to degrade the polymer, say absorbable suture, suture thread, or biodegradable suture. Absorbable suture means when those suture threads are used for stitching purpose after surgery or for the purpose of healing a wound. So, it comes in contact with the body fluid. Now, if it is non-absorbable, non-biodegradable suture, after wound healing, there is a necessity of removal of that suture thread from the body. That is a painful affair. Now, if there is some absorbable suture which will slowly be absorbed within the body fluid and the degradation products will be excreted from the body slowly, then that removal step can be avoided. Okay. So, say internal operation suppose within the body, after operation it is actually closed and stitched. Now, that is done by tissue adhesive or suture. Now, if the suture is used, again if it is not absorbable, then it is to be again removed after the wound is healed. So, for those purpose we have to see what polymer we should select depending on the requirement. There are certain requirements, say tissue engineering scaffold for hard tissue replacement, say total hip joint replacement or knee replacement or finger replacement, finger joint replacement, anything. So, for hard tissue means bone replacement. What we do? We make a scaffold from calcium, synthetic calcium hydroxyapatite, calcium hydroxyapatite. You have heard this name, calcium hydroxy. It is a bone mineral. It is present in bone, normal bone. Now, if a skeleton or a scaffold is made, porous scaffold is made from this thing, or a composite is made from this calcium hydroxyapatite or bioglass or calcium phosphate using collagen as the matrix, this is the reinforcement, this is the matrix. So, a composite will be formed, a composite. Now, that composite can be used for replacement of bones, or else we can make scaffold from calcium porous scaffold from calcium hydroxyapatite. Today people are making bioceramics, porous bioceramics. Now, after replacement what happens? Native bone formation will be there, means it will help osteoconductivity. It will help in osteoconductivity and native bone will be formed and slowly those pores will be filled. Okay. Or if you take some composite made from collagen, calcium hydroxide, calcium phosphate or some other polymer, say polyhydroxybutyrate. Polyhydroxybutyrate or valerate, polyhydroxy or polyhydroxy valerate. These are actually bacterial polysaccharide. Bacterial polysaccharide produced by bacteria. It is a polymer, polysaccharide polymer. It is a biodegradable polymer, or chitin or chitosan, okay, or chitosan. Now, these can be used as matrix with calcium phosphate or calcium hydroxyapatite or even bioactive glass. I mentioned earlier bioactive glass. So, what will happen? These polysaccharides or collagen or other biodegradable, say polylactic acid, polyglycolic acid, polylactic co-glycolic acid copolymer. Now, these biodegradable polymers with this calcium phosphate or bioactive materials or calcium hydroxyapatite, that will form a composite that can be used for replacement of bone. Slowly these polymers will degrade within the body, create some space. In those spaces the native bone tissue will be formed. Okay. So, after surgery, say after 6 months of surgery or 5 months of surgery, one can find that the artificial part which was replaced, which was put inside the body, that is no longer present there, is almost original bone. Native bone has been formed there. So, that can help in healing of some damage, bone damage, or affected or diseased bone, all these things can be replaced. There are some today people are using nanomaterials. Yesterday there was a lecture in our center. They have shown some bioceramic, nano bioceramic made from nanocellulose and nano bioceramic particles. Okay. So, we can use these polymers which must be compatible with the host system. That means, we must try to find out some similarity of these polymers with the polymers present in the host tissue. Yes, please.
Native bond will be formed, or the replaced bone will act like a native bone?
No. If it is biodegradable, that polymer will degrade slowly, some space will be created, and there native bone tissue formation starts, and it will be filled by the native bone.
This composite works as a catalyst for making the native bonds.
It's not possible.
No catalyst. Okay. So, this is
how it induced the regeneration of bone.
By normal your physiological system, physiological process. You see, if there is some injury on a tissue, if there is no external attack by external agency, there will be normal healing, regeneration of tissues by its own system.
So that is limited for a certain age.
Do not go to that complications at this stage, but it occurs. Certain age, you see, it has been found that at the age of 90 years, fractured bone is repaired. How? It happens. It happens due to falling in the bathroom at the your old age. People gets their bone fractured. After plastering and some treatment, it is cured. So, it is not that so long a system is living, there is all the moment there is tissue growth, tissue formation, either bone tissue or soft tissue. You cannot say at the old age this tissue formation is totally stopped. No, it is there. Rate may be slower, the situation may be little difficult, but it is there. So, what we see, polymers are used in biomedical field. It is again a vast subject. It is a vast subject. I am giving you little exposure, introduction to you, so that if anywhere it is necessary, you can explore. Then ceramics are also used, metals are also used, titanium, vanadium, aluminum, alloy. These are used for total hip prosthesis today. Ceramics are also used. Now, since you are material scientists, you should not keep aside this concept of bonding. Always you should think in terms of chemical bonding present in those materials and devices. Primary bonding and secondary bonding, nature of bonding, nature of primary bonding, nature of secondary bonding, and total quantity of primary and secondary bonding present. So, you should take care of that properties, requirement, biocompatibility. That means, the external or foreign system, foreign body should be acceptable or should be accepted without any hazard, without any toxic effect in the body. If it is not, if there is cell proliferation, cell growth, tissue growth, etc., all those things in presence of those foreign bodies, then we can say this material is biocompatible. And there is methods of testing also to test the biocompatibility of a material, be it polymer, be it ceramic, be it metal. There are tests are available, standard test methods are there. That means, what we do, what is normally done in order to test the biocompatibility, you take a specimen of the material, put in a culture media, okay, containing certain cells. If in presence of that specimen the cells continue to grow or proliferate, populate, we say there is no toxic effect. So, we can say in one way it is biocompatible, that is, this is in vitro test. So, by in vitro test we can say whether this material is biocompatible or not. Also, this is not complete, you have to go through also in vivo test. You have to put that specimen inside a living system, say animals. You have to go to animal for animal testing. That means, you put this thing in the animal, you see the habits, behavior, change of habits, behavior, their food and other your body temperature, etc., all these things. If those remains unaffected, it can be certified that yes, it is acceptable by the living system, biological system, biocompatible. So, this is the second step success. Third step success is, if we want to apply in humans, then in the third step it should be applied in human system. Again, some monitoring system should be there. Through some monitoring system, if there is no adverse effect by putting this thing inside the body, then it can be recommended as a suitable biocompatible material, biocompatible device which can be used. There are physical properties as well as chemical properties. You know what are the physical properties, you know, chemical properties, you know, mechanical properties, strength aspect, tensile strength, modulus, elongation at break, hardness, their compression set, and sometimes your dynamic properties, dynamic mechanical properties, etc. Then thermal stability also are to be considered at body temperature. At body temperature, whether those materials and devices are stable or not. Sometimes some materials need electrical conduction, so electrical properties as well as optical properties. Then stability and degradation, how long they are stable. If there is slow degradation in presence of body fluid, so you have to test it through simulated body fluid, and you have to see if there is any degradation. You have to take out the specimen, you have to analyze the specimen, and you have to analyze the surrounding medium, say fluid or tissue, if there is any degradation product or not. If there is some degradation product, you have to see what are the degradation products, are those toxic or not. Those things have to be evaluated for these materials. So, acceptance of an implant by surrounding tissues and by the body as a whole can tell you about the biocompatibility. The implant should be compatible with tissues in terms of mechanical, chemical, surface and pharmacological properties. Simply, it is the ability of the implant material to perform with an appropriate host response in a specific applications. Host response, response of the host after accepting or after putting this thing, behavior of the host. Mechanical properties, say cortical bone, cancellous bone, enamel, dentin materials, say modulus, their modulus, tensile strength. Okay. Then mechanical properties of soft tissues, those are the mechanical properties of hard tissues. Here these are the mechanical properties of soft tissues, cartilage, ligament, tendon, skin, arterial tissue in two directions, intraocular lens. So, you see the requirement of this indicates the requirement of mechanical properties of the materials which are to be used for such biomedical devices. Now, there one can apply the nanotechnology in the fabrication, during the fabrication of such devices. Okay. So, you see this is the tensile strength, this is the modulus, this is the tensile strength. Oh, this is wrong actually, this should be gigapascal, this should be gigapascal, not megapascal, modulus is higher. Anyway, then mechanical properties of typical ceramic and metallic biomaterials. You can compare the tensile strength and modulus of some metals and ceramics. Okay. You compare, metals are stronger, tensile strengths are more in case of metals, and hydroxyapatite, you see hydroxyapatite, this is a bone mineral, but alumina, a very hard ceramics, alumina, zirconia, all the hard ceramics. So, their properties are higher than those of hydroxyapatite. Here is a problem. Okay. If you want to replace any part of the body with such foreign materials like metals and ceramics, your properties matching is very difficult to obtain, is not it? You look at the properties, they are very strong. So, there is a mismatch of these properties. Then you look at the polymers, the mechanical properties, say modulus and tensile strength, these properties are close to those of soft tissues, and mechanical properties of some composites can be brought very close to those of hard tissues. That is possible with polymers. Okay. And that can be tailored again following this nanotechnology. Nanotechnology, I mean you have to follow certain process so that nano dimensional reinforcement is possible to form. Say think of cellulose, cotton. If we want to make nano nano dimensional cotton fiber, how we can make? You know, cotton contains cellulose molecules. Cotton in the cellulose molecules in the cotton fiber highly crystalline, and that is the reason for its insolubility, infusibility. Cotton, which contains anhydroglucose unit rings, anhydroglucose units. Those anhydroglucose units unite one after the other to form a cellulose molecule, and there is extensive intermolecular hydrogen bonding between cellulose molecules through hydroxyl group, one primary hydroxyl group and two secondary hydroxyl groups are there. Okay. By virtue of that hydrogen bonding between those hydroxyl groups, there is intermolecular hydrogen bonds present in cotton, cellulosic materials. Now, if you want to make nanofiber out of this cotton fiber, it is difficult in the sense it is not soluble in a solvent, it is not fusible. You know what to do? Now, if you can find a technique to break the hydrogen bonds, intermolecular hydrogen bonds, then you can separate the cellulose molecules, means your cellulose molecules from each other. Then only it is possible to form nano cellulosic fiber. and once these nanocellulosic fibers are formed, these hydroxyl groups will be exposed or opened, hydrogen bonds will be broken. Again there is every tendency of reformation of hydrogen bonds. That is the problem. That means, when you make some nanoparticle or particle in nano dimension, eventually there will be recombination to form the aggregate. That means, it becomes difficult to maintain the nano dimension. So, if it is possible to prepare this nano unit in situ, that means, if immediately some your third thing comes between two units, nano units, then that will prevent their recombination. Then only you can get dispersion of nano units in a matrix. So, in case of cellulose, say what you can do? You have to break the hydrogen bonds. How to break the hydrogen bonds? There are two ways, thermal and chemical ways. Now, if you apply thermal energy, that should be in such a amount, so that it only reaches the energy of that hydrogen bond which can break, is not it? You can use some chemical energy, some chemical materials, chemical compound, chemical agent. If it can provide that energy close to that of hydrogen bond breaking energy, then that can break, and eventually you have to disperse those nanofibers, preventing recombination. So, we can imagine one thing, a cotton fiber or any other natural fiber, that is a bundle of fibrils. A fiber is a bundle of fibrils. Fibrils are having smaller diameter than fiber. Okay? A fiber can be considered as a bundle of fibrils. So, your job would be to separate those fibrils from a fiber by breaking intermolecular hydrogen bonds. Now, again each fibril can have large number of cellulose molecules in a bundle. So, fibrils can be bundle of cellulose molecules and fiber is bundle of fibrils. So, in each step you have to break the hydrogen bonds. Now, there is a technique called steam explosion technique. What is that steam explosion technique? You take these fibers in an autoclave, you put these fibers in water, or take a dispersion of these fibers in water, take that dispersion in an autoclave, close the autoclave, okay, start heating, means it is, if it is electrically heated, put on the switch. So, it will increase the temperature. So, that water will vaporize and form steam. Okay. If you go on increasing the temperature, so you will have steam at higher temperature, a superheated steam will be there. So, in that condition what will happen? These water molecules will go in between those fibrils as well as in between these cellulose molecules, and the energy available over there is sufficient to break those hydrogen bonds. So, water molecule can be penetrated in between those fibrils and cellulose molecules. Then what you have to do is suddenly open the autoclave. This is at high pressure. Okay. You go for sudden release of pressure. What will happen? So, inside the fiber there are water molecules in between fibrils and cellulose molecules. Okay. So, on sudden release of pressure there will be some bursting effect. So, water molecules in between the fibrils, those are under pressure. So, outside that when the pressure is released, so that fiber will be burst out, we will say this your fibrils will be separated, fibers will be separated. This is one way, and that will remain dispersed. Water is there. So, then that will, those cellulose molecules will try to form hydrogen bond with water molecules, not with another cellulose molecule. So, intermolecular hydrogen bonds will be broken, but intermolecular cellulose hydrogen bonds will be broken, but some hydrogen bonds between cellulose and water will be formed. Is it clear? So, what we will get? We can get a dispersion of nanocellulose fibers in water. This is one way. In another way, you can go for ultrasound treatment. If you expose this system, says that slurry or dispersion to ultrasonic your energy environment. So, by this sonication, these hydrogen bonds can be broken, depends on what frequency of ultrasonic energy at what frequency you are using, you are applying. So, this way you can make nanofibers. So, these techniques are being followed everywhere, wherever one technique is suitable. The selection of any technique is up to you, which you can really handle. So, this way one can get nanofibers. So, here you see, if you can make the fibers from these polymers in nano dimensions, or some fillers, say clay, nano clay, etc. So, if you can disperse in these polymers, so you can make nano devices, nano composite devices for biomedical application, and there will be intimate contact between the fiber and the matrix. If there is intimate contact between the fiber and the matrix, then only you can get the nano effect. What happens in macroscopic composites? In macroscopic composites, this reinforcement either in the particulate form or in the fiber form, those are in bigger dimension. So, total interface area between the reinforcement and the matrix is less there. Okay. Now, if you think of say 5 gram of material, if you can break it to nano dimension, you can calculate the total amount of surface area of the particles available will be available in the nano dimension. Say 10 nanometers, suppose 10 nanometers, 5 gram of 10 nanometers of material, you can calculate the surface area, considering, assuming it as spherical particle. Okay. Then you compare this total surface area with that of 40 gram of the same material having say micron dimension. You will find that the surface area available from that nano, the smaller amount of nanomaterial will be higher than that one. So, what it is giving? It is giving that more surface or interface for contact between the matrix and the fiber. So, we can get fruitfully the nano effect, nano composite. So, this is the concept. Only thing, we have to make the experimental, we have to develop the experimental setup, then only we can get the success, otherwise it is difficult. Implant devices, scaffolds for tissue engineering. Now, to talk about scaffold for tissue engineering. Now, today people are making scaffolds from nanofibers. Yes, people are making silk nanofibers. Silk is a natural fiber. Now, that is actually dissolved in a suitable solvent, then by nano spinning process or by electro spinning process. So, that solution of that silk in a suitable solvent is spun through narrow opening, then electric field is applied across this spun fiber. say fibers are drawn, fibers your filaments are coming down from the nano that spinneret, and some electric field is applied. So, at that high electric field, the fiber dimension becomes very thin. That helps actually that your application of electric field at high voltage helps in getting very thin fiber. So, people are making conducting polymer nanofibers by that technique. I have seen that setup, that device. People are making conducting polymer nanofiber. People are making your silk nanofibers, collagen nanofibers, protein nanofibers by this technique, and those are used for making scaffolds. Those scaffolds are used for tissue engineering. That means, scaffold is a support, on which these tissues are grown, cells are grown to get a proper tissue in a proper shape and dimension. In the last class I was telling myocardial tissue construction by cell sheet engineering, myocardial tissue construction by cell sheet engineering without using any scaffold, with the help of that stimuli responsive polymer or thermo responsive polymer. But you can use some scaffold for tissue engineering. So, that scaffold can be a porous support, of course, that should be a porous support, or a network, or a hydrogel or gel materials, where ample space is available for tissue growth, tissue accommodation there. And ultimately, if the scaffold is biodegradable, then by slow biodegradation, ultimately we can get a native tissue. So, hydrogels in regenerative medicine, bones and joints, vascular grafts, heart valves, tendon and muscle, drug delivery devices, contact lens, sutures, and so many large number of applications can be mentioned. Now, polymers in healthcare, I have told many things. Now, look, the examples of polymers: polymethyl methacrylate. These acrylate polymers, these acrylic polymers, because of their excellent refractive index, those are used in optical devices. So, contact lens, rigid contact lens, earlier for rigid contact lens manufacture or preparation, PMMA was the ideal material, but it creates certain problems of hydrophobicity and lack of porosity, all these things, barrier properties, because it should permeate water molecules, sodium chloride and other things. So, today's optical your contact lens is being made of this acrylate polymers, but not PMMA or a copolymer of other monomer with methyl methacrylate. Have you heard this name? Polyhema, polyhydroxyethyl methacrylate, polyhydroxyethyl methacrylate. You know methyl methacrylate. This is methyl methacrylate. Now, in case of polyhema, this group is replaced by hydroxyethyl group. Hydroxyethyl group. Now, you see, by placing that hydroxyethyl group, it increases the bulkiness of this portion as well as some CH2 group, additional carbon, that regulates the hydrophilicity and hydrophobicity as well as packing of the molecular packing. So, what we need? We need some space to passage for the passage of this water and other salts, sodium chloride, other things, tears, suppose tears. Not only that, it should maintain a proper flexibility, or it should be it should remain moist all the time. It should not get dry. Now, if it is hydrophobic in nature, if it is not, if it cannot invite water molecules, cannot like water molecules, then that will cause irritation in the eye. That is why today copolymers, I do not know the composition, this is not disclosed, I do not know, but copolymer of polyhema, hema with MMA, hema, or sometimes here in place of hydroxyethyl group, there may be other alkyl group also. Then polyamides, polylactic acid, okay. PVC for blood bag. You have seen blood bag for collection of blood, that is made of PVC, although it is banned today. That is flexible PVC, plasticized PVC. It is banned globally, but in India nobody bothers. So, we are using this flexible plasticized PVC. The problem is, it gives a life to the blood to say for 30 days only at 4 degrees Celsius. 30 days life is available. And again, we have, it has been found that within 30 days there is rupture of your blood components due to the presence of plasticizer over there. Now, plasticizers are ester plasticizers. Now, those ester plasticizers hydrolyze to form monoester. Those monoesters actually come out from this PVC, plasticized PVC film and mix with the blood, that contaminates the blood, that it brings some toxicity to the blood. That is why this plasticized PVC is banned, still people are using. In our lab, we have developed some PVC film for blood bag without using any plasticizer. Okay. Catheters, you have seen catheters, flexible tubes, soft tubes, transparent tubes, IV system, those are made of PVC. Okay. Vascular prosthesis you see, say mesh or nets or fabrics made of polyethylene terephthalate fiber. This is called Dacron fiber, Dacron fiber, trade name of polyethylene terephthalate, Dacron fiber, that is used for making vascular, that means your veins or arteries, along with some fluorinated polymer fiber, fluoropolymer fibers. If anybody of you is interested, literature is available, you can go through, you can see. There is a course on biomaterials, you can take that course also in order to get in details. Polyurethanes, although these polyurethanes are made from what? Polyurethanes are diisocyanates and diol. You know, isocyanates are highly toxic. Now, once these isocyanate groups react with hydroxyl group, forms urethane bond. Once this urethane bond is formed, it is non-toxic. Because of that, these polyurethanes are used in biomedical implants also. spandex fibers, polyurethane fibers, those are used in biomedical field. So, those are used in catheters, cardiac pumps, sometimes your oxygenator, blood oxygenator membranes. Silicones, silicone is an inert material, silicone rubber. That silicone is used for making cosmetic surgery, say breast replacement, breast implant, or cosmetics other, say or nose or replacement, nose surgery, or finger joint or fingers, artificial fingers, these are made from these silicones. Okay. Polyphenol alcohol and copolymers for making hydrogels for drug delivery purposes, and poly N isopropyl acrylamide, NIPAM polymer. In the last class I told you that is used for making thermoresponsive controlled drug delivery devices, as well as for bioconjugates for separation and other things. These are various photographs of composites made from carbon fiber and epoxy, carbon fiber polyether ether ketone, you see, for joints, bone plates made of composite materials. These are commercially available today, these composites, high performance composites. Although these are used in aerospace, used in automobiles for structural items, here also you see those are used in as bone materials. Screws, you can have metal screws. Now, metal screws are being replaced today by these composite screws made from carbon fiber and PIC, polyether ether ketone. Hip joint, hip prosthesis, again the same carbon fiber PIC, injection molded composite are being used today. So, it will take some minimum time to explain these things. Next class I will see to cover these portions. Okay. Thank you.